The workflow consists of growing cells at strictly controlled environmental conditions, rapidly chilling them using melting water ice, and fixing with pre-optimized concentration of formaldehyde and incubation time. The fixation reaction is then quenched using excess of primary amino group containing compounds such as glycine or tris. Cells are collected, washed, and can be stored frozen. Fixed cells can be mechanically or chemically lysed, and the lyse is used for separation of the ribosomal fractions based on their molecular size, sedimentation properties, or affinity markers. The approach results in versatile material usable for immuno-assisted purification and detection, enrichment of complexes containing specific factors in electron microscopy. Upon cross-linked deblocking, RNA amplification or hybridization-based analyses, RNA sequencing, and mass spectrometry become possible.
Set up a 1 liter yeast culture in an orbital shaker with a starting optical density no more than 0.05 absorbance units at 600 nanometers in yeast peptone and dextrose media at 30 degrees Celsius. Set up a preparative centrifuge with compatible rotor and centrifuge bottles for pelleting the liquid suspension of yeast cells at 5000 G at 4 degrees Celsius. Keep record of the optical density of the growing cells. It must be between 0.6 to 0.8 absorbance units at 600 nanometers at the time of collection if the exponential growth phase is of an interest. Once the cells are ready, set up an icebox inside the chemical fume hood with a beaker containing 250 grams of clean crushed water ice. Ensure you also have 25 milliliter stripettes and freshly purchased methanol stabilized